I'd like to thank the organizers for the invitation to be part of this Congress and to talk about reproducibility analytics for qualification of microphysiological systems and interpreting biologically, clinically relevant heterogeneity for precision medicine. So as in vitro models have evolved from simple 2D single-cell static models to multi-cell organoids and fluidic structure biomimetic platforms, managing the increased amounts of data and metadata has become more challenging, as has assessing the reproducibility of the model performance. Now that we are at the stage where these multi-complex in vitro models are being submitted for FDA and regulatory qualification as drug development tools, it's critical to effectively manage these large data sets in order to properly assess the reproducibility of the platform, as well as to apply them for precision medicine. So with the 21st Century Cures Act, which required the establishment of qualification process for drug development tools, reproducibility is becoming really important. Drug development tool is a method, material, or measure that aid in drug development for regulatory review. A DDT can be relied upon for specific interpretation and application for its proposed context of use once it's qualified. And the context of use is the specific use of the DDT for development. And a qualified DDT not only helps optimize drug development and evaluation, but also can be included in IND, NDA, and BLA submissions without having to reconfirm the suitability of that model for that application. And qualification requires demonstrating the reliability and accuracy of the DDT for its context of use, and reproducibility is central to reliability. Now, reproducibility in clinical research is a major challenge. Up to 88% of preclinical research is not reproducible, and even 60% of researchers can't reproduce their own results. So I think there are a number of reasons for the lack of reproducibility, and I think you can read those there. But I think what's most important are the ones that I'm highlighting here is, the statistical analyses, and the standardization of the data and the metadata, and the absence of actually having access to the data and metadata for the analyses is critical. So in assessing reproducibility, it requires a systematic approach. It requires the inter-study reproducibility, requires repeating measurements under identical conditions in the same study. It requires repeating studies on different days or in different labs. It requires managing study data and metadata, including detailed experimental protocols, standard operating procedures, and detailed description of how the data are analyzed, and also having ready access to all the metadata and data for this analysis. Now, an essential tool for tracking reproducibility is an integrated analytics platform. Now, the analytics platform needs to capture details about how the model is set up, the MPS model and device, the cell samples that are used, very detailed, including patient information from the cell sources, how the experiment is actually set up, and how the assays and the measurements are used, what assay methods are used, what protocols, and how it's being analyzed. And these metadata are compiled in a database which integrate now the study data. And these data can now be put into different or various analytic modules to help analyze the data. And then if clinical data are available, this can also be integrated with this in vitro data to now start putting together predictive models for precision medicine. Now, to standardize the workflow or the reproducibility analysis, we've developed the Pittsburgh Reproducibility Protocol, which is a standardized workflow to evaluate reproducibility and identify heterogeneity in these complex models. And it starts with coefficient variation, which is a measure of dispersion of points about the mean and the precision of the response. And we've established for our internal use these metrics of it's got to be at least less than 15% in order for it to be reproducible based on CV. If it's less than 5%, it's an excellent reproducibility. If it's between 5 and 15%, it's an acceptable reproducibility. However, when it exceeds 15%, the CV, we then look at the analysis of variance, which basically tests that the population means are equal. And we can assign reproducibility status here based on the p-value. If it's less than 0.05, then it's consistent with perhaps not being reproducible. If it's greater than 0.05, it's not consistent with it being not reproducible. For multiple time point metrics, we use the interclass correlation coefficient, which measures the reliability of results for categorical data. It reflects the degree of correlation and agreement between measures, and since it is a correlation coefficient, those which come closer to 1 shows a much better correlation. And so we established that 0.8, ICC of 0.8 or above is an excellent reproducible result. Between 0.2 and 0.8 is acceptable, and if it's less than 0.2, then we consider that to be poor reproducibility. Now, to illustrate the prep in practice, I'm going to walk through an analysis that we ran assessing the reproducibility of our liver MPS platform. And the disease that we are addressing is metabolic dysfunction-associated steatotic liver disease, which is a complex, heterogeneous, and progressive disease, as you may know. It affects 30% of the world's population, and it can lead all the way up to hepatocellular carcinoma. The model we developed is the liver CNS microphysiological systems model, or LAMPS. It's a structured biomimetic, which we're developing as a drug development tool. It's constructed with the 4 liver cell types. It has a physiological architecture of the liver CNS. It's amenable to inducing different disease states, including MASLD stages of 0 to 3. And it can be applied for ADME-tox studies, studying liver disease progression, as well as being a precision medicine platform for patient if we use patient iPSC-derived liver cells. And in the upper right on the table, we have developed a number of media formulations for inducing different stages of MASLD in our model. And these are based on the blood chemistries of patients at different stages of the disease. Now, the CRISPR reproducibility protocol can evaluate the reproducibility on 3 different levels. Now, the first is the absolute signal level, which is the amount of analyte that's produced or intensity of a signal. And in this example here, we're showing the induction of steatosis by lipid volumes under either the disease state, which is EMS, the blue bars, or in a non-disease state, which is NF, or the orange bars. And if you look at the CV across, the results across 5 different studies for these 2 different conditions, it's a bit high. It's above our 15% cutoff, but de novo is suggesting that, hey, this is not consistent with it not being reproducible. In cases where the absolute signals are not reproducible, we look at the relative signal levels. And this is an analyte signal normalized to internal controls. And in this case, we're looking in panel B at the secretion of collagen 1A1 under the disease and normal conditions. And you can see the statistics suggest that that's not reproducible. However, we're more interested in the relative secretion of the collagen 1A1 in the disease state relative to the non-disease state. And when you take that ratio and normalize it to the NF, you can see that it is quite reproducible across the studies with a CV of less than 10%. Now, there are cases where that is not reproducible either. And then we look at the trends in the signal, or the consistency in signal changes. And in this case, we're looking at IL-6 secretion in the disease state, and even under the normalized conditions to NF, you can see that the reproducibility across 4 different studies is poor. However, what we do note is that in all 4 studies, it's always increased in the disease state relative to the non-disease state. Now, while this is not a good metric for drug screening, for example, It is useful for model development because at least it tells you you're going in the right direction. You just need to optimize some of the conditions to make these more reproducible. And then finally, looking at the inter-class correlation coefficient, which looks at the reproducibility and trends over time. Here we're looking on the left at the secretion of collagen 1A1 over time in 5 different studies. And the ICC suggests that is not reproducible. However, if you normalize the data to their respective medians for each of the studies, you can see they overlap quite nicely on the right-hand side. And so what this says is that the trend for the secretion of the collagen 1A1 is highly reproducible across different studies in our model. So now, applying this to precision medicine, Maslow's patient heterogeneity complicates drug development and selecting patient cohorts for clinical trials and patient treatments. And the patient heterogeneity in Maslow stems from different genetics, different lifestyle, different environments, and different comorbidities. And even there are 2 drugs that are conditionally approved for Maslow, but they have a very low response rate in the general population. And so the heterogeneity, this type of heterogeneity, necessitates the need for a precision medicine platform to optimize patient cohorts and individuals who will have a high probability of responding to the drugs. So we've developed our platform for precision medicine, the LAMPS platform. And first we needed to establish the technical reproducibility. And like I said earlier, that's when you're looking at all identical components under identical conditions, shown in column A here. And once it's been established that it's been highly reproducible, you can then start applying that to different cell samples for different patients, for example. And what this will show you is the reproducibility or the biological heterogeneity. And you will have some patients whose reproducibility across themselves or among themselves would be good, but that reproducibility will be different from another group of patients. And so this will allow you then to start putting patients together into different cohorts that have similar heterogeneity metrics for drug discovery, clinical trials, and so forth. So now, to illustrate this, we've constructed the lamps using hepatocytes from different patients, and in this case, we're inducing steatosis in our lamps platform. Now, here I'm showing 31 studies that we've performed with these different patient samples, and the overall reproducibility among the patients is poor, 56% CV, and the p-value is also not very good. But if we then separate the studies based on the disease condition or disease stage, we see in the top you have a normal fasting or a non-disease state, and on the bottom this is the early metabolic syndrome or disease state. The CVs are getting better. They're still not good, but nonetheless segregating out based on the stage of disease is showing better concordance. Separation of these studies further based on this patient cell genetics for PNPLA3 variant vastly improved the reproducibility of response in the induction of steatosis in the model. This suggests that a difference in the response to the induction of steatosis between GG variant, which has been associated with increased susceptibility, Maslow's, relative to the wild type CC variant. Now we can further segregate them down into studies of just the individual patients, where the same patient was run in multiple studies, and you can see that the reproducibility is very good looking at the individual patients. What this tells us is that the model is technically reproducible. It also says that we can use these metrics and our model to segregate out different patient samples and then use them and put them in different cohorts. For example, here you would want to put all the GG variants into one cohort and the CC variants in another cohort if you're going to run into a clinical trial. So just to summarize, complex in vitro models are important tools for drug development, and they are being qualified for regulatory qualification. Establishing reproducibility is necessary to critically interpret these experimental results. And identifying and understanding the biological and clinical heterogeneity is the foundation for precision medicine. But this has to be deconvolved from the technical variability in order to draw valid conclusions. We've developed the Pittsburgh Reproducibility Protocol as a standard approach for systematically assessing reproducibility. And the PrEP, along with the study metadata, enables the distinguishing between the technical variability and the biological heterogeneity for application for precision medicine. Then I just wanted to acknowledge the group that's been involved in the production and development of the models as well as the metrics, and I'd be happy to address any questions.